Replication, DNA helicase unwinds a double-stranded DNA to expose the two single-stranded template strands. Proteins known as single-strand DNA binding proteins, or SSBs, stabilize the strands. Since the template strands are anti-parallel and new DNA is made in the 5' to 3' direction, the process of chain elongation occurs differently on the two strands. For example, let's look at the replication fork in an E. coli chromosome. First, DNA primase synthesizes a short RNA primer in the 5' to 3' direction. Next, DNA polymerase III adds DNA nucleotides to the RNA primer in the 5' to 3' direction. This new DNA is called the leading strand because it is being made in the same direction as the movement of the replication fork. On the top template strand, DNA primase synthesizes a short RNA primer in the 5' to 3' direction. Then, DNA polymerase III adds DNA nucleotides to the RNA primer in the 5' to 3' direction. This new DNA is called the lagging strand because it is being made in the opposite direction as the movement of the replication fork. The short segment of DNA produced along the top template strand is called an Okazaki fragment. Meanwhile, new nucleotides are added to the leading strand, so that the leading strand is synthesized continuously. On the lagging strand template, however, replication can only proceed in the 5' to 3' direction. For DNA replication to continue on that strand, a new RNA primer is synthesized by primase near the replication fork. The primer is then elongated by the action of DNA polymerase III to produce a second Okazaki fragment. Thus, the lagging strand is synthesized discontinuously, producing Okazaki fragments separated by gaps. Even as synthesis of a new Okazaki fragment begins, the first two Okazaki fragments are joined together into a continuous strand. First, another DNA polymerase I, removes the RNA primer and replaces it with DNA. After the primer nucleotides have been replaced, a single-stranded gap still exists between adjacent nucleotides. An enzyme known as DNA ligase closes these gaps. The action of DNA ligase produces a longer, continuous DNA strand. The process repeats as the DNA continues to unwind. Because one new DNA strand is synthesized continuously and the other is synthesized discontinuously, this model is called the semi-discontinuous model for DNA synthesis.